So maybe we should, oh yeah, we have. Let's start off. We also have some questions that we've been accumulating to ask you in case everyone is too shy. But we do have a question here in front.
Thank you very much. Anton Bogars from Institut Pasteur. I have a question for Arnaud. A lot has been said about what the next pandemic could be, and you all have talked a lot about the impact of climate change on those next pandemics. If you had to make a guess, would you say this next pandemic could be related to a respiratory disease or more to a vector-borne disease or a zoonotic disease?
Let's say a guess would be difficult because there are so many possibilities, and as the slide I showed was telling that it can come really from anywhere, and we have not been able so far to anticipate the place and timing of the next emergence. But if you are talking about viruses that would lead to a worldwide pandemic with a very significant impact, my guess would go to respiratory viruses because they have this route of transmission that makes everyone at risk. And provided they have sufficient lethality, I would put a threshold around 1 per 1,000, for instance. And as I was mentioning, if they become transmissible before the onset of symptoms, which is the case for influenza viruses so far, and has been the case for SARS-CoV-2 compared to its predecessor, which were transmissible only 5 days after onset of symptoms. But when you get those three criteria, you get a worldwide pandemic of really significant impact. So for me, the real threat for a pandemic of the scale of what we had with COVID-19 would go to respiratory viruses. And arboviruses are important because we do see that they are moving to now in terms of geographical boundaries, in terms of intensity. Last year in France, we had for the first time 67 cases of dengue transmitted on the French territory, from imported cases. You may have seen in the media recently 4 cases of malaria in Florida. These sort of things will happen more and more. Currently, it remains limited for the temperate zone, but unfortunately, in tropical countries, these have really a major impact. And indeed, arboviruses would be pretty close on my list. I would just mention and finish with this because I believe there are many questions, but just imagine what would happen if yellow fever was moving to Asia. There are in Asia Aedes aegypti that can transmit the yellow fever virus. There are increasing links between China and Africa. And during the last epidemic of yellow fever in Angola in 2016, there were 11 cases of yellow fever diagnosed in China from Chinese coming back to China. Had any of them been bitten by Aedes aegypti, difficult to predict whether there would have been a sustained transmission, and there are questions around that. But this is the sort of things we would not want to see happening.
Are there any questions? Yeah, we have one on the side here.
Pushkar Sharma from National Institute of Immunology, New Delhi. The question relates to the TRP1 talk. My question was, if I understood correctly, you are conditionally knocking down the channels in these mice and doing those studies. So I'm looking from the point of view of receptor sensitization and desensitization. So have you looked at second messengers such as adrenaline and others during exercise in these animals, and what impact do they have on receptor activation and so on and so forth?
So if I understand you correctly, if we looked at receptors that are changed based on acclimation and exercise?
Or modification of TRP1 channels by signaling pathways and their contribution to the process.
So we have started to look at this because, I'm a neurophysiologist and the underpinnings of the changes are really interesting to us. So we have started by sequencing these cells, RNA sequencing, to see what messages go up and down. We haven't looked so much yet at modifications of these ion channels because we reasoned the time scale we look at is really long. It's not a classic plasticity mechanism that happens over seconds, minutes, and so on. This is very long. So it would be at least plausible to see that expression changes of certain receptors. And we see this, certain ion channels that are known to sort of be involved in firing rates and in excitability of cells are changed. And we have now one particular one, a sodium channel, which we clearly can also functionally implicate in this. And there are probably many changes going on in these cells, and that our transcription profile actually shows. So what does that mean? I guess you hint indirectly at it. Can this be utilized? Can one pharmacologically modulate these channels to sort of prime organisms, mammals, for being ready for heat? I think that future will have to show how specific certain receptors are in these areas, if one can really do this. This is really early days in this regard, and we haven't tried anything in that direction. But hope would be yes, that one can somehow artificially sort of stimulate specific neuron sets that can then help to better cope with heat.
So Jan, on that, can Andy, the epidemiologist, and Jan, the physiologist, find the biomarker? You spoke about a biomarker for heat exposure or for heat resilience. So could maybe Andy and Jan, could you talk about how you could work together to collaborate on this as a transdisciplinary, to talk about these biomarkers that we really do need to observe and study heat exposure and change over time?
Yeah, I mean, I think there are a number of different challenges, really. One is trying to understand how to monitor the exposures to extreme heat and the exposures that are physiologically and pathologically relevant. And, of course, there has been work on topics such as heat shock proteins and so on. But in our very preliminary results from studies in West Africa, these seem to be quite insensitive to the kind of outcomes that we're interested in. So they're really in extreme conditions. And what we really need is very sensitive biomarkers, which will tell us when the exposure is sufficient to be beginning to interfere with kind of physiological capacity to adapt. And I suspect that that will be rather different in different populations. So, for example, different amongst the elderly, different, the other issue of pregnant women and the fetus and so on probably would need different biomarkers there. So I think it's a fascinating area. I think we would need to look at different populations if we're going to do that. Some of them in relatively unacclimatized populations. So, for example, in Europe, when we get a heat wave, these are exposing people who are vulnerable, but they're relatively unacclimatized. Whereas if you look at, say, Asia and much of Africa, the populations are acclimatized, but nevertheless exposed to much more extreme heat. And so I think we'd need quite a sophisticated approach, looking at different populations, different health outcomes, different levels of acclimatization. But it would be fascinating to collaborate.
Maybe, yeah, just to add on this, I mean, with the animal model, it might be different in humans per se, it might be different in different populations. We would have potentially the advantage to have like controlled populations, acclimatized versus non-acclimatized, and then look at surrogates, blood samples, and see what has changed. Yeah, that could be done. I would still like to know what one would looking for, right? One can always screen now metabolomics, transcriptomics, proteins. So maybe if I would have to guess and start somewhere, I think actually metabolic parameters change very dramatically. We see this also if we put mice at these warm temperatures, they lose weight. And maybe along those lines, I totally agree, heat shock proteins is probably too crude, and they would also need to be in the bloodstream, right? I mean, heat shock proteins work, many of them at least, in the cells. So other biomarkers are probably better suited in that regard. But maybe, yeah, one could expand not only looking at proteins, but maybe at metabolic changes and looking in the bloodstream might be.
So we have a new grant to be funded here. And we can throw in the mitochondrial people as well. I can see there's a nice synergy working here. Are there any more questions? We have another one. Okay, we'll do that. Are you listening? Yeah, I think it's very interesting. We're looking at some of the, in hot areas in South Africa, whether there's an increased incidence of intimate partner violence, and that might be a proxy or a biomarker of early lack of resilience to heat or as it changes over time. So we are doing a study looking at the role of intimate partner violence and heat in areas in our country where it's very hot. Are there any questions? Oh, you can, on your side there.
Just,
Can you hear me now? Okay. So just say congratulations and thanks for absolutely mind-boggling talks and ideas. Hello, Mike, again. I would like to start two questions. One is to combine a little bit discussion which we had during the panel with Mona Niemer and Marcia McNutt about what this age of biology may mean in the future and what kind of limitations and frames we need to take in mind to be able actually to profit from it rather than not. Jan, I found your story absolutely fascinating. And you said, we tried this to do artificially, right? Which means you succeeded, actually, to stimulate and to understand what the neurons did. The question of audience was maybe also meant to be, suppose, somewhere in the near future, what nears can be, this could be done somehow without doing any harm at the regions where people, humans, suffer from heat stress. Now, how far are we from that? And what kind of ethical and other limitations we would have? It goes straight to the discussion we had this morning. And second question goes to Sophie. Sophie, in HSSPO we have 5 countries which do have an enormous historical heritage from the indigenous population. Canada, United States, New Zealand, when I just came from recently, Australia, and Norway with the Sami community. This is, of course, a small example of what can and should be done. Would you ever advise what we can do, combining those 5, 6 countries with their nations, actually to do a more systemic dive into the possibilities which the indigenous knowledge in the life science, biology can help us? Thank you.
So do you want to start, Sophie?
So can you repeat the last part? And so we have 5 continents, and you want to give...
Can work together. So if we have all this biodiversity and this movement into looking at natural medications, is there a role of working together and collaborating to evaluate this at scale?
Absolutely. I think, so for now we are very local. It's even Alberta, it's Lethbridge. But these plants can basically serve anyone, and we are looking for collaboration because we are looking for very particular topics. As I said, poultry for me, anti-cancer for Roy, but they can serve many things. And we also can use plants from other countries. So we were collaborating with Pierre Fabre. They had, this very huge plant extract library, and we can also try to test them. And so, absolutely, because there is the antibiotic resistance issue is not. It's not a Canadian problem. It's a problem that is everywhere. So I think the more we are to try to address this issue, the better it will be, and the faster it will go. So plans from other countries for sure, if you have some.
So the response about the question raised around the issues around temperature, the ethics of evaluating this data, and any responses around the issues around the heat exposure and the questions that were raised.
Yeah, I mean, one was at the time frame, right? These are really early days and really basic scientists with animal models at this stage, and I couldn't even predict how long it would take. What I can say is there's several levels, right? First, is already touched upon by the question from back there, we would have to find a way to be able to manipulate this without putting artificial switches into these neurons. So either pharmacologically, what I could also imagine is some sort of external extracranial stimulation, talking about basic science at this point, right? Not humans, but model organisms. I think that could be something which is been done in different contexts, pain treatments, and others that you stimulate from outside and can activate certain areas of the brain. And there's more and more research going in this direction as well, also in humans, in completely different contexts. I could imagine that this could also be potentially applied here. Fantasizing now, not knowing when and how, but it could be. If we find a good way to do this, then the next question is, what side effects would there be, right? The hypothalamus controls many different aspects, not only temperature. And a challenge would likely be the cells that do different types of, mediate different types of physiology are intermingled in the hypothalamus. So it's locally not, you cannot really easily, at least we don't know so far, dissect this tiny area is only for this and this is that. It's likely not. It's sort of very much intermingled. Nevertheless, maybe, so cardiology changes, metabolic changes, circadian cycle, things like that are controlled by the hypothalamus. How this all would be affected by such a treatment, I can only speculate at this point, right? That's one would have to see if one finds a good model that would potentially also be applicable to humans to see what in these parameters is changed in humans. So ethics. Yeah, that would depend on what we will find with these types of side effects, I guess, right? So that would be even more of a speculation at this point, what that would all entail. One thing that makes me hopeful is energy metabolism and these acclimation aspects actually work together in some ways. I mean, and I think we all know if we acclimatize, we usually are less hungry, and we see this with the mice. They eat less, they lose weight, which is principally also beneficial, or can be beneficial. So in that sense, if one does sort of such a stimulation, one hope would be that the concerted effect is sort of going in a similar direction and being positive as an overall effect, but that one would have to see.
Yes, I have a naive question in terms of biomarkers for extreme heat and heat adaptation, etc. I recall some research some time ago out of Finland on changes in some parameters, physiologic parameters after sauna exposure. I mean, that would be quite ethical, I think, to carry out. I'm just wondering whether you have any, comments or any information that would be relevant from such approaches.
Yeah, thanks. I mean, I am aware that there's been quite a lot of work from Finland and other countries of short-term exposures under sauna conditions, including potentially vulnerable groups like pregnant women and so on. I think one also has to ask oneself to what extent that's representative of some of the challenges that we're going to experience, particularly, say, in low-income settings where you've got people with very different nutritional, nutritional challenges, if you like, exposed to extreme heat conditions. Also, it doesn't tell us very much about what happens when people have to have sustained physical activity in extreme heat, which is the kind of problem we're facing in parts of Africa, with the populations that we're working with there. So I think certainly these kind of physiological studies can be useful, just like physiological studies in heat chambers can be useful. But the problem with heat chambers is, again, most of those studies have been done in fit, young, healthy volunteers from Europe, North America. As far as I'm aware, there's possibly none or very few heat chambers in Africa, for example. So we don't really know how those populations.
So in the mining industry, so
the mining,
miners,
South African mining did,
in that they to acclimatize to the heat in the underground.
Yeah,
miners go through that whole heat, that whole process.
But they're often, they're mainly studies in minors, I think. Again, not so many perhaps vulnerable people like older people or pregnant women or whatever. But yeah, no, you're right to remind us of that, yeah.
Are there any, we have a question there.
Yep, thank you. I also wanted to talk a little bit about ethical issues, particularly linking climate change and health, and thinking about the planetary boundaries way of the view of looking at this. So we've, I think, Andy, you talked about nature-based solutions in terms of planting trees and mangroves and so on. And so nature-based solutions are one, nice way that people like to think that we can deal with the urgent problems we have today. The other way of dealing with it are technology-based solutions, right? And so I wonder, from a biological perspective, when do nature-based solutions become technology-based solutions? We heard, from Marcia this morning about gene drives, about synthetic biology. It's no longer science fiction to think about, generating viruses that can help us to generating good viruses that can help us deal with bad viruses, or creating synthetic organisms that can maybe help us deal with climate change. So would you define those as nature-based solutions because we're using nature to help us, or do you define these as technology-based solutions, which are more typically, considered, engineering issues, right? So how do you see this nature-based versus technology-based view of dealing with planetary boundaries, and do you think it's sort of dangerous to silo nature versus technology, and we need somehow to think about it in a more holistic way?
Thanks. Yes, it's a great question. Just one general point about nature-based solutions: we shouldn't see them as alternatives to phasing out fossil fuels. There is a danger at the moment that they're being emphasized a lot, including by interests who don't necessarily want to tackle the fundamental problems of phasing out fossil fuels. But if they're considered to be a part of the spectrum of potential solutions, then I think that's totally valid. So whether it's a technology or a nature-based system, I think it depends a bit on the types of nature-based solutions you're talking about. So there are some which are really the sort of type 1, as they're sometimes called, which are really about protecting current ecosystems. So they're really about capitalizing on the current system, the integrity of the current ecosystem, and protecting that. Then there are those that are moderately modified by humans, like agroforestry. And then those that are almost entirely new. So, for example, green roofs. I mean, that's a human developed ecosystem, if you like. And I think that we could see as a kind of technology. There may be different types of, I'm sure there are, different types of green roofs which will have different effects on local climate, on the internal environment of the building in which they're based, on biodiversity and so on. They may have different health effects. So I think it's a spectrum, and we shouldn't be too, we should accept that there's going to be a spectrum of different types of nature-based solutions. And we may be able to learn from the natural solutions about technologies that could be employed through some of these totally new and human, synthetic, if you like, ecosystems. So I think there should be an interplay between those people who are studying the kind of ecology and then those who are studying potentially new technologies which can help to adapt to or mitigate climate change and protect biodiversity.
To even be more provocative and maybe get to some of the other questions, it's the gain of function or the mosquito adaptation. So is that, what about using these kind of technologies, gain of function or mosquito adaptation, which is, kind of a, this manipulation of the environment, and what are the risks of doing things like that?
With the mosquitoes, I think the interesting part with Wolbachia, it's a bacteria that already infects naturally 20% of insects. So it's not that we are introducing something completely new there. And again, it targets a very specific species of mosquitoes, because you are going to release Aedes-infected Wolbachia, and therefore you will target the Aedes that process. There are other approaches which are using genetically modified mosquitoes, some also with a gene-drive component that would therefore select it preferentially at the future generations. But currently there are some moratorium on this type of research, at least at the development of field trials, because we are not able yet to fully anticipate the consequences of the introduction of those sorts of things.
What about manipulating viruses? So we want to understand coronavirus a bit more.
So this is a different set of questions, which is where do you put the trade-off between trying to understand better what sort of mutations would make a virus more transmissible, and therefore you would be able to better monitor them in the surveillance that you could do in wild and farming animals, and the risk that by doing these gain-of-function studies, you create a virus that ultimately ends in the population. There had already been a discussion on that when some researchers from the Netherlands and Japan were trying to understand better how H5N1 could become transmissible. H5N1 is this avian influenza that is highly lethal. I mean, 50% of humans who have been infected died, half of them, but fortunately is not transmissible among humans. And some researchers were trying to find out what would be the mutation that would make it transmissible among humans, and that has been stopped after some discussion, because we felt like it would be just too dangerous to have these viral modifications in the laboratory infecting some lab workers and getting into population. And I think it was wise. And the recent episode of the COVID-19, everybody has it in mind. So we probably will never know what exactly happened. Most people believe that there has not been changes of the virus, SARS-CoV-2, that led to the one that became responsible for the pandemic. What people are currently debating is whether that virus was present in a lab in China and leaked from the lab, but it had been collected from a bat, or whether it came from the animal market. Currently, I think that the pendulum has moved more to the market, to the lab, but I think this is something that we will probably never know. But I think we realized through this episode the risk associated with this type of research. And I think that fortunately there will be a lot of attention put to what sort of experiments can be done and in which type of labs, because we realize that there is risk attached to it.
Yeah, so scrutiny of labs, spillage, documenting illnesses amongst lab workers, all of that. That gets us to the ethics of mitochondrial so maybe expand a little. We got 1 minute, 2 minutes. Sure. And you mentioned your work on the mitochondrial donation, for want of a better word. So maybe you could just expand in the last minute on that.
And I think it's a really good segue because this is legislation was required for a new IVF procedure. First, it was achieved in the UK. The House of Lords approved it. It was a very strong body of evidence through the Wellcome Trust, Eli Lilly Foundation, pushing it with great spokespeople from scientists working together. And the community got around it, and they supported it as well. So that was really community-driven, but politicians really drove that. That led the ability for our organisation, our Mitochondrial Foundation in Australia, to start doing the headway in Australia to try and get an IVF procedure approved for legislation there. That was, again, a ground up, a lot of discussion, scientists going and meeting politicians, talking around that, and eventually, actually, our right-leaning government at the time were the ones who actually put it forward. And it's often, I think, what you need to have is the right-leaning governments to actually make the decision about something that's going to be very strong and go for it, because then it's going to be accepted. And so that got through in legislation. What it means is that if it's achieved through a clinical trial, is that mothers will no longer pass their mitochondrial DNA down the line, their maternal line, to their children. They will be now passing the mitochondrial DNA from another donor for the rest of the generations that are going through from that maternal line. It's mitochondrial DNA. It's only about what it makes is a few proteins that's involved in the energy generation. It's not about blue eyes, intelligence. It's just the machines of life.
How
and
you
they're
breathe.
good machines, right? And so this is the idea of correction. So having that conversation around how to best work through approaches that actually make a lot of sense. Of course, we had a lot of resistance from some religious lobbies, obviously, but again, the politicians in the end agreed to this trial. So now we have to see whether it's actually going to be achieved.
That's going to be fascinating. I don't know if my co-chair has anything to say, but before I hand it over to him, just to remind the people who are going to be loading who's speaking in the next session, please go and load your slides during the break. And we need to come back at 3. Okay, thanks.
So I'd like to thank our speakers. I think I'm not going to ask other questions because we are running over time. So thank you all for your illuminating talks and this discussion, which I think really illustrated the connection between climate change and how our health is influenced by this climate change and what kind of solution we should be building in the coming years. So thank you very much. Thank you to the audience for your questions and the discussion. Thank you.